Most life on Earth follows a rule so basic we rarely question it. If you want to get big, you must become many. One cell divides. Those cells specialize. Some contract, some sense, some signal. They assemble into tissues, tissues into organs, organs into systems. Control emerges from hierarchy. Intelligence emerges from wiring. Strength emerges from structure. Size, we are told, demands complexity. That story feels so natural that we mistake it for a law. But scattered across Earth's oceans, ponds, and sediments are organisms that never followed it. They grew large without dividing. They coordinated without organs. They learned without neurons. They are one. And the deeper we look at them, the more unsettling the implication becomes. Life did not need to break itself apart to become powerful. It only needed to engineer itself differently. Among all known single cells, Valonia ventricosa is the most visually shocking. While most cells survive by stretching into thin threads or spreading into sheets, Valonia does the opposite. It condenses its existence into a single, massive, gleaming sphere. It is often called the largest discrete single cell on Earth because it refuses the usual tricks. It does not elongate to stay alive. It does not fragment into compartments. It stays whole. If a typical human cell were scaled up to the size of a person, a single Valonia sphere would be the size of an aircraft carrier. Valonia has solved a problem that biology usually avoids entirely: structural engineering at macroscopic scale inside a single membrane. This is not growth. This is construction. The secret begins with its wall. Its wall is a composite material built from cellulose microfibrils ranged in carefully controlled layers. This architecture is identical in principle to plywood or carbon fiber composites. Each layer resists force in a different direction. Together, they prevent tearing, buckling, and splitting. The result is anisotropic strength, resistance to stress from nearly any direction. Inside that hole, Valonia maintains an internal pressure of up to 4 atmospheres, comparable to or greater than the pressure inside a car tire. Valonia survives because of geometry. It is nearly spherical, the most efficient shape for distributing pressure evenly. Instead of bones, it uses hydrostatic tension. Pressure pushes outward, the wall pushes back. The cell becomes a living pressure vessel. But pressure alone would not be enough without its most elegant trick. Valonia is mostly empty. Up to 95% of its internal volume is a central vacuole filled with water. The living cytoplasm is compressed into a thin layer just beneath the wall. Functionally, its metabolism operates into dimensions instead of 3. Diffusion remains microscopic even though the organism itself is macroscopic. Valonia breaks the square cube law by becoming hollow. It is a giant that lives like a microbe, while Valonia grows its own armor. Spiculosophin Oceana proves that a single cell can be a master of external engineering. It is an architect that builds its own fortress. It roams the ocean floor for spicules, tiny discarded glass needles from dead sponges. It gathers these needles and glues them together with its own proteins to build a rigid, towering stalk. By building this tower, the cell escapes the boundary layer of the seafloor where water is still. It gains the high ground, allowing it to harpoon passing prey that would otherwise be out of reach. It uses a skeleton it didn't have to grow to perform a professional role usually reserved for complex, multicellular corals. If Valonia forces us to rethink structure, Physarum polycephalum forces us to rethink intelligence. Physarum is a single-celled slime mold that can spread across several square meters, forming a pulsating network of living tubes. Inside those tubes, cytoplasm flows back and forth in rhythmic waves known as shuttle streaming. These flows are driven by actin and myosin, the same proteins that power muscle contraction in animals. The physics does the thinking. This is why Physarum can solve optimization problems that humans approach with mathematics. In a famous experiment, researchers placed oat flakes on a surface in the exact positions of cities around Tokyo. Within about 26 hours, the slime mold built a network connecting them. The structure closely matched the real Tokyo rail system, balancing efficiency, cost, and redundancy. Physarum remembers without a brain. As it moves, it leaves behind a trail of extracellular slime. The mold avoids its own old trails, treating them as explored territory. The environment becomes memory. It can even share experience. When two Physarum individuals touch, they fuse. Knowledge is not transmitted. It is mixed. It does not think about the world. It thinks with it. If Physarum dissolves the boundary between body and mind, Warnoia dissolves the boundary between cell and organ. Warnoia is a single-celled dinoflagellate that contains an eye, not a light-sensitive spot. An eye? Inside its membrane sits the ocelloid, a structure so complex that scientists once assumed it belonged to a multicellular animal the cell had eaten. It includes a cornea-like outer layer, a lens formed from a repurposed mitochondrion, and a retina-like pigment cup derived from a plastid. Warnoia hunts transparent prey in open water. Evidence suggests its oceloid can detect polarized light, revealing distortions invisible to most organisms. There is no brain. The oceloid is physically linked to its stinging apparatus. Light in, action out. Vision becomes a trigger, not a perception. Warnoia does not process images. It responds to optics. If a single cell can build a telescope for an eye, it can also build a compass for a brain. To navigate the three-dimensional darkness, Loxoids uses the Muller organ, a dedicated internal gravity sensor. Inside the cell sits a tiny mineral stone, a crystal of barium sulfate, suspended by delicate membranes in a fluid-filled chamber. Because the stone is heavy, gravity always pulls it down. As the cell tilts or flips, the stone shifts, physically triggering the cell's internal sensors. This organ allows the cell to perform gravitaxis, piloting itself with the precision of a cockpit instrument. In the oxygen pore ponds where Loxoids lives, swimming the wrong way for even a few minutes is fatal. The Muller organ acts as an internal level, telling the cell exactly which way is up so it can find oxygen in total darkness. Acetobularia reveals something colder, memory without presence. This single-celled alga grows into a delicate stalk topped with a lace-like cap. Its nucleus sits at the base. If the cap is removed, it regrows. If the nucleus is removed, it still regrows. The instructions were sent long ago as stable messenger RNA molecules, anchored in the cytoskeleton. The cell runs on cache data. Growth continues without a designer. Dialmargarita magnifica pushes scale beyond what bacteria were ever supposed to achieve. Discovered in 2022, this single bacterium grows up to 2 centimeters long. It survives by decentralizing its genome to hundreds of thousands of membrane-bound pouches called pepins. Every region of the cell can read instructions locally. It is hollow, redundant, and chemically autonomous. A bacterium built like infrastructure. Spirostomum ambigu improves unity is strength under violence. This giant ciliate can contract 60% of its body length in less than 5 milliseconds, reaching accelerations that would liquefy any animal. It survives because nothing inside it can collide. No organs, no skeleton, just fluid and flexibility. Unity is armor. Across all of these organisms, a pattern emerges. They do not lack sophistication. They lack fragmentation. They sense without nerves. They remember without synapses. They compute without symbols. They move without muscles. They build without hands. They pilot without brains. They endure without organs. They show that intelligence, resilience, and power do not require becoming many. Sometimes, the most robust design is the one that never breaks itself apart. Life did not climb a single ladder toward complexity. It explored multiple paths. And one of the most radical paths was this: to remain one, to engineer unity, to let physics do the thinking, to let structure replace hierarchy. In a world that demanded division, these organisms refused, and they survived anyway. If you enjoyed this video, subscribe, like and share to see more content like this.